So I'll be presenting our ongoing project, environmental impact of diluted bitumen, or DILBIT. And the main players here are presented on the main slide. People from across the GSC, INRS, and McGill. So some background. I gave this last year, but just for those of you who didn't presentation last year. So in 2019, crude bitumen production from Alberta oil sands region totaled around 3 million barrels per day, compared to around 0.5 million barrels per day for conventional crude. And of course, that's set to increase in the near future. To transport bitumen via pipeline, it is blended with lighter hydrocarbons, yielding a less viscous diluted bitumen, DILBIT. So bitumen is a very highly degraded form of petroleum that's very viscous, so they need to blend it with lighter material to put it into pipelines. And although DILBIT is generally considered safer than other means of transporting DILBIT by pipeline is generally safer than other means of transporting DILBIT. Pipeline ruptures have occurred. For example, a major one that happened in the Kalamazoo River in the States a few years ago. And so DILBIT spills may behave differently than conventional crude spills, but our understanding of the environmental impact of DILBIT in freshwater environments is limited, particularly in groundwater systems. So the activities that fall under our project, whose goal is to better understand the relationships between geochemistry, hydrology, microbiology, and toxicology during natural attenuation of DILBIT using controlled spill experiments, both the lab and the field. And our activities fall under two main categories: shallow groundwater systems and riverine and lacustrine systems. And today I'm going to focus only on large column experiments and large tank experiments that we carried out at the INRS Heavy Labs, or Labalure facility here in Quebec City. So I showed some data from this last year and the year before. So we carried out some unsaturated stone spill experiments using large columns, using sand we obtained from the Galt Nature Reserve in Mont-Saint-Hilaire near Montreal. It's operated by McGill. And so we had columns set up that contained that we had above ground spills of diluted bitumen, and compared that to a heavy conventional crude and a control. And we monitored the water and the soil over time. So in terms of the water samples, this was the data that I discussed last year. To maintain, spring-autumn recharge conditions, to mimic spring-autumn recharge conditions, we had a temperature-controlled room that was set at 9 degrees Celsius. We added water, which was averaged to precipitation over the August to November time period, 6.25 liters per week. We adjusted the pH to reflect the slightly acidified precipitation in the Montreal region, and we measured a suite of parameters over time, including TOC, BTEX, pHs, acid extracted organics, that is containing naphthenic acids, major elements and metals, and we did fathead mineral toxicity analysis. But I'm not going to show any of that this week. This year, I'm going to focus on the soil cores. So this work made up Leah Mindorf's MSc thesis. She's based at McGill, and now she's working in the Delta Lab at the GSC here in Quebec. So we collected 10 cores over 15 weeks, and we collected cores from 3 different sections. You can see here the top, middle, and bottom sections, corresponding roughly to 45 centimeters in depth. We filled up the holes with bentonite after collecting each core in order to maintain, to not disturb the hydraulic conductivity that large holes would have caused. And we carried a suite of analysis: total petroleum hydrocarbons, TPHs, phospholipid fatty acids, PLFAs. I'll get to those in a second. The next slide. Carbon-13 analysis of PLFAs, C14 analysis of PLFAs. And as Paul described, we carry out the same type of microbial community analysis, 16S ribosomal RNA analysis. And we have some ongoing analysis that have been delayed from COVID and other reasons, which were pH concentrations and compound-specific hydrogen and carbon analysis of pHs. And those are being carried out as we speak. So PLFAs. What are phospholipid fatty acids? Well, they're biomarkers for the active microbial population. They're found in the cell membranes of microbes, and You can see here when you analyze them, you're analyzing the individual lipids, and their carbon-13 and carbon-14 analysis can provide unparalleled insight into carbon sources. So you're looking at basically what microbes are eating directly. Look at the carbon content. So here's the carbon-13 values of the PLFAs from the soil cores combined together. So here we have the conventional crude in red, the control in green, the dilbit in blue. And these dotted lines correspond to the carbon-13 content of the particular source of carbon, the soil, dilbit, or conventional crude. And you can see here that there's not a lot of differences between the three main types of large column experiments. There's some differences in the PLFAs within the groups, but not between the types of experiments. And in general, aerobic transformation of these carbon sources, it produces about a 2 per mil or 3 per mil offset in the carbon-13 signature of PLFA. So you can see there's not a lot of difference. This data by itself is not particularly useful to understand any processes. But when you look at carbon-14 contents, and this is in order to get enough material for C14 analysis, we had to combine the entire extract of PLFAs. And so here we have here on the graph scale is the delta notation of carbon-14. Minus 1,000 per mil is the value that you find in fossil fuels. It does not contain any carbon-14. Anything older than around 50,000 years old will not have carbon-14, so it gives you a value of minus 1,000. More modern organic carbon, of course, is up here, and the value of carbon-14 in the soil organic matter from our column experiments is this dotted line right here. And the important thing is to look at the control, the triangles. Here we have very little change in the control, and we have either the combined depths or the different depths of the soil cores over time. But as you can see in the conventional crude and in the dilbit cores, you see a shift. You see more depleted values in the PLFAs over time that you don't see in the control cores. You don't see any trends between the different depths or between the dilbit or conventional crude, but you definitely see a difference between the control, which is showing you unambiguously that there is definite microbial uptake of the two petroleum types over time in the cores. And so here's a microbial community analysis at the genus level. And the genus are dominated by bacteria. The relative abundances are expressed as percentage. And the group boxes, the different boxes group column treatments, top axis, and depths, y-axis. And as you can see here, there were significant increases in the relative abundance of Polymonas bacteria over time in the columns in both dilbit and conventional crude, but not in the control. And so Polymonas have been previously identified as potential dilbit degraders. And you can see here that we see this evidence also in our experiments. Okay, so moving on to the large tank experiment to mimic a controlled dilbit spill in the saturated zone. Here we are filling a large tank that's approximately, it's 3 meters length by 1.5 meters width by 2 meters height. And we obtained the sand this time from a local quarry near Quebec City. And here we are filling it up and installing piezometers at different vertical depths across a source zone, which I'll get to in the next slide. The sand was filled up to 1.75 meters depth. So the source zone in this experiment was, we had a different method to make the source zone as opposed to just pouring it on top. Because you want to maintain a source zone that was buried under the surface, and also we want to minimize buildup of free product. So we decided to target a concentration of 15,000 milligrams per kilogram, or ppm, of dilbit. We mixed this in, we homogenized it, and we buried it using this wooden box to a depth of around 86 centimeters to around 9 centimeters below the surface. And we had a flow rate of around 1,000 liters of, we used deionized water. 1,000 liters per day was pumped into the system at a gradient. hydraulic gradient of 0.00173 to simulate a groundwater flow. Okay. And here's another perspective of the tank. The source zone is here, flow direction's here, and so the site of upstream water injection and the outflow were separated by the sand by a vertical perforated plate, which you can kind of see here near the outflow. And here's a piezometer nest, 3 different piezometer nests, each at, and we had horizontal, sorry, and vertical nests, top, middle, and bottom, that covered the entire vertical length of the tank. Okay, so I'm not showing any results from that. I have to wait till next year's presentation. Thank you very much.